Doing that, I'll go ahead and welcome everyone to the HIV and Global Health Rounds at the Antiviral Research Center. We have the pleasure of having Dr. Gabriel Wagner present today on updates in HIV prevention from virtual CROI 2021. Just a little bit about Dr. Wagner, pulling off his website here. He did his undergraduate work and received a BA in biochemistry, biology, and French at Case Western University. He did his medicine training at University of Toledo College of Medicine. He was at the Cleveland Clinic for his internal medicine residency, and then was here at UCSD for his infectious disease fellowship. He was the winner of the Young Investigator Award for CROI back in 2012 to 2014. And he conducts translational HIV research and is active in practice in infectious disease and also at the Owen Clinic as an HIV primary care physician. So I'm glad to introduce my colleague, Gabe Wagner. So take it away.
Thank you, Dan. Can you all hear me?
Yep. And by the way, I'll monitor your chat, and if there's anything, I'll let you know.
Okay, sounds good. Hi, everyone. Thanks for being here. I'll try. It's always challenging to do these because there's so much that is covered at CROI regarding HIV prevention. It's really impossible to cram it all in 1 hour and make it cogent. So I'll try my best. I have no disclosures. So I'll be borrowing heavily from the talks by Jean-Michel Molina from University of Paris. He gave a great talk trying to summarize the state of affairs in the program committee workshop for new investigators and trainees, as well as Marina Kasky and Linda Gale Becker, who gave plenary sessions. And there was a great interactive session. And I'm saying I'm putting these out here, so if you all get the chance to do so, and when it becomes available, to really enjoy these sessions. It was a great interactive panel on how long-acting agents, how efficacy can be translated to effectiveness, with Francois Venter from South Africa, Nitaya Panupak from Thailand, Charles Flexner from Hopkins, and Hyman Scott from San Francisco Department of Public Health. So this is, I'll be talking about basically the topical, oral, and parenteral forms of PrEP, approved and not approved yet. And I won't have the chance to go into vaccines very much. Okay, so the dapivirine vaginal ring, this was the field regarding prevention using microbicides kind of started with the seminal, the Caprisa 004 trial 10 years ago, but then since has moved into the ring now, the dapivirine vaginal ring. These were seminal studies, mirror studies that were done published in 2016. The ring study in partnership with the International Partnership for Microbicides, and the Aspire study in partnership with the Microbicide Trials Network, and the randomized double-blind studies using the dapivirine vaginal ring versus placebo. This is a flexible silicone matrix, and the ring has 25 milligrams of dapivirine, and it's self-inserted every month, so every 4 weeks. And basically, on the left, the ring study, you can see there are 2,000 young women, South Africa and Uganda, 31% reduction in HIV incidence. And on the right, the Aspire study, 2,600 women in sub-Saharan Africa, there was a reduction in HIV incidence of 27% and 37% after exclusion of 2 sites that had poor adherence to the study protocols. So these were followed with open label extension studies, and these, the DREAM study and the HOPE study, and they, the self-report adherence to the ring and acceptance to the ring was good, and the reduction in the incidence increased 39% and 62% respectively. And here is a report from a user in Cape Town, 20 years old, and I'll let you go ahead and read it. So clearly, this has a direct impact on the users. So next steps with the dapivirine vaginal rings, this was the European Medicines Agency approved the ring in July 2020 for women in high HIV burden areas where oral PrEP is not available. And the WHO recommended it just a couple of months ago for the dapivirine vaginal ring as a new choice for HIV prevention for women at substantial risk of HIV infection. And this probably opens the door for wide use in Sub-Saharan Africa, and probably will be approved here in the US before long. So more studies are ongoing, which is good. We need more diverse cohorts. So the REACH study in South Africa, Uganda, and Zimbabwe will be looking at adolescent girls and young women. The DELIVER study in South Africa, Uganda, Malawi, and Zimbabwe will be looking at pregnant women. And the Be Protected study will be looking at breastfeeding women. And there's increasingly the use of multi-purpose technologies for contraception and HIV prevention. So rings combining dapivirine and the contraceptive 11 or gestrel. So now this is the first study that I'll be talking about that was presented at CROI. This is fresh new data. This was presented by Albert Liu over at the San Francisco Department of Public Health. And here they looked at 3-month dapivirine vaginal rings. So you may remember that the seminal studies were done with 1-month dapivirine rings. Here they're looking at just phase 1, safety and acceptability of the 3-month dapivirine vaginal rings. This was MTN 036, IPM 047, a phase 1, 3-arm randomized 1-to-1-to-1 trial to either the standard 1-month 25 milligram dapivirine ring, or two different dosages of higher concentration dapivirine worn every 13 weeks, or worn continuously for 13 weeks. So this 100 milligram or 200 milligram. 49 HIV uninfected women and those assigned female at birth. It was interesting because included in the cohort was 1 trans man. Not a lot of data on those folks. 2 study sites, UAB and San Francisco Department of Public Health. And basically what they found is that the majority of adverse events were mild, 79%, or moderate, 21%. There was 1 grade 3 adverse event, which was probably unrelated. And the table here shows the concentrations of the 100 milligram vaginal ring in green and 200 milligram vaginal ring in blue. 100 milligram vaginal ring in green and 200 in blue. Across the columns are plasma, cervical vaginal fluid, and cervical tissue at different days. And basically what you saw was an increase in the plasma concentration anywhere from 1.3 to 1.9 using the higher concentration 3-month rings, and in cervical vaginal fluid 1.5 to 2.9 increased concentration. The most participants liked the rings, median of 8 on a 10-point Likert scale. And most were likely to use the ring again if effective. That was scored as a median of 9 on a 10-point Likert scale. So it looks pretty good, and it'll go forward probably soon on phase 2 trials. Okay, so regarding oral PrEP, Jean-Michel Molina, the seminal study on on-demand PrEP was the EPAIR-GAY study from the ANRS in France. This was sex-driven immediate PrEP, 2 tablets 2 to 24 hours before sex, 1 tablet 24 hours later, and 1 tablet 48 hours after first intake. And this was data already published. This was the EPAIR-GAY study in 2015, basically showing they had a mean follow-up of 12 months, and 16 subjects were infected, 14 in the placebo arm, and 2 in the TDF-FTC arm. and the incidence resulting in an incidence in the placebo arm of 6.6 per 100 person years and an incidence of 0.9 per 100 person years in the TDF-FTC arm, resulting in an effectiveness of 86% in relative reduction in the incidence of HIV, which was 100% when people took the medication. So here they presented, Dr. Molina presented an update on the kind of continuation of the IPERGAY study, the Prevenir ANRS study. This is PrEP with daily or on-demand TDF-FTC among MSM. This was an open-label prospective cohort study in the Paris region. So here they gave the participants the choice. Participants opted for either daily or on-demand PrEP and could switch regimens. So this was open label. It couldn't be randomized. It couldn't be blind. Sorry. There were 3,000 HIV negative, HIV risk adults, and you can see there the study protocol and the endpoint. The primary endpoint was showing a 15% reduction in new HIV diagnoses among MSM, followed every 3 months, and the risk reduction measures that you see there. So unfortunately, the primary objectives could not be assessed because there couldn't be a proper surveillance of the estimate of the number of new infections in 2019 or 2020. So that could not be assessed. So instead, he presented some of the other secondary characteristics, secondary endpoints in the study. So the study at this point in the analysis has 3,067, 98.5% MSM, and 49.5% of these were on demand, so nearly half and half. So the on-demand arm was shown to have significantly fewer condomless sex acts and significantly fewer sex partners at baseline. Only 3 patients discontinued permanent PrEP due to gastrointestinal adverse events, 2 in the daily arm and 1 on the with the on-demand arm, which is interesting. You know, you might expect that the daily regimen might cause more GI side effects, but it didn't. All grade ALT elevation, the incidence was high actually in both arms, 22.2 in the daily arm and 18.3 in the on-demand arm. However, when looking at grade 3 ALT elevations, the incidence was low in both arms. Again, low incidence of creatinine clearance that was low in both arms, with a low creatinine clearance in both arms. And both arms did have a high incidence of bacterial and viral STIs and hepatitis C. These were data that were presented by Karen Hoover from the CDC. These are trends looking at TDF-FTC and FTAF prescriptions in the US from 2014 through 2020. So this was interesting, actually. You know, the FTAF was approved for use in October 2019. We really don't know what the prescription trends have looked like since then. So I think this is the first attempt to kind of look at that. So they used the, I don't know how to say this, IQVIA or IQVIA. You can tell that I'm not in the VPN. Using this information, they looked at the prescriptions from 2014 through now. And what you can see on the graph, so this is starting 2014. This is the number of persons prescribed PrEP. Steady increase, a nice increase actually in the uptake of PrEP usage. And then in October of 2019 is when FTAF was approved. You can see a stark increase in the prescription of FTAF and a decrease in the prescription of TDF-FTC. So 36.3% of new PrEP users after October 2019 were prescribed FTAF, and 28.5% of 228,000 TDF-FTD users were switched to FTAF from TDF-FTC. PrEP users who switched to FTAF were more likely to be older, male, Hispanic, Latino, and also to live, I think it said anywhere but the Northeast or something strange like that. And less likely to be cash payers, which that might not come as any surprise to any of us, given insurance coverage of Descovy. Next, this was an interesting science spotlight, also from the CDC, Dr. Henney, presenting PrEP trends in 48 urban jurisdictions by federal funding. So here they looked at, again, using the IQVIA longitudinal prescription database, looking at PrEP users and looking at providers. And they looked at 48 urban jurisdictions that were included in the ending the HIV epidemic. So, and here they stratified again by federal funding. So federally qualified health centers versus non-federally qualified health centers. And what you can see in this table is that there's been a nice steady increase across the board from 2014 in the amount of PrEP usage and that reported PrEP users and PrEP providers. And the rate, though, the estimated annual percent change was higher in the FQHC, in the federally qualified health centers, which is good, which is encouraging. And they said that, hopefully we can continue to see these kinds of trends, and those services can then bleed out into the larger communities. This was another interesting study that was presented by Patrick Sullivan. So their group at Emory, I'm a fan because one of my areas of interest is mHealth and how it can be used for PrEP, for prevention. So they had been working on this app, and here they presented data on their randomized controlled trial. This is the app, they call it M-Cubed. So this is built on social cognitive theory, and a forerunner version of the app had demonstrated acceptability and usability. So the app can do a couple of different things. It can do prevention messages. So this is prevention messages surrounding condoms, PrEP, STIs, and HIV cares, and it comes in two forms: infographics and short videos. You can see an example of the infographic in the picture there. Unsure about your HIV status? One way to take charge of health is to get tested, and you can click on find a testing location near you. It also provides core services, so established testing and frequency. You can see in the panel in the middle there, you can choose a testing strategy, which is nice and convenient. It can also it provides risk screening and service locators, as well as information about prevention. And also commodity ordering, so bundling condoms and lubricants, for example, that do well with certain condoms, as well as HIV self-test kits and STI specimen self-collection test kits. So this was a randomized one-to-one trial, randomized one-to-one to immediate onset of app usage versus delayed onset of app access. I think it was 6 or 9 months. And the enrollment was from Atlanta, Detroit, and New York City, 1,220 overall. And the study group was stratified into 3 different groups. It was stratified into higher risk HIV negative, lower risk HIV negative, and those living with HIV. And what you can see in the prevalence ratio on the right there, in the bottom right, is that for the higher risk HIV negative group, the proportion of HIV testing was significantly higher, and there was an increase in PrEP use. And these were at the time of sampling. The time of sampling was 3 months after use of the app. There was no significant effects for the lower risk HIV negative group, and there was no significant effects for those living with HIV. So that seemed interesting and promising as well.
Gabe, can I ask a quick question?
Yes, sure.
On that slide, was it true there was 6 months follow-up though, and there was no effect in the higher risk HIV negatives, or am I reading that wrong?
The sampling was done at 3 months.
Okay.
That's when no. What is shown there on in the diagram on the bottom right?
Yeah.
No.
Okay.
Yeah. It it is interesting that they didn't see any significant effects in the low risk HIV negative, but perhaps you know, something those who are using the app more and the services more have some level of risk perception more than the other groups. So now jumping into novel PrEP agents, and we'll begin with Islatravir. This is a new potent and long-acting antiretroviral agent. This is previously published data. So by way of background, nucleoside reverse transcriptase translocation inhibitor, a little hard to say, has potent antiviral activity with low in vitro IC50 of 1.5 nanomolar and activity against strains that are resistant to NRTIs. And so a single dose of oral Islatravir was associated with a reduction in plasma HIV RNA without emergence of viral resistance. And you can see in the plot there on the right, this is a change from baseline HIV RNA log and over time, and different dosing strategies, and different dosing concentrations of Islatravir. And there was a robust viral load decline associated with Islatravir triphosphate concentrations as low as 0.05 picomolar per million cells. So that's the threshold that they defined for activity. So one of the nice things about Islatravir, as several of the other agents that were discussed that I think are soon to come, is that they have very long half-lives. And so Islatravir triphosphate has a half-life of 120 hours in healthy adults. And concentrations in rectal and vaginal tissue are similar to concentrations in cells, which is fantastic. And in addition to this, it protected against HIV infection in a rectal challenge rhesus macaque model. So all these things make it very attractive for PrEP. So these were data that were not presented at CROI, but at the R4P conference that took place last month. And this is presented by Sharon Hillier. This was a double-blind, randomized, placebo-controlled study of monthly oral Islatravir for PrEP. So this is taking Islatravir orally once a month. And they the observed mean Islatravir triphosphate concentration time profile was measured in cells and overlaid on population PK model simulated median Islatravir triphosphate concentrations. And here they looked at two different dosing strategies, 60 milligrams once a month and 120 milligrams once a month. And what you can see is that the Islatravir triphosphate trough concentrations remained above the PK threshold of 0.05 picomolar in both instances. So Merck plans to go forward. They have a clinical development program using 60 milligrams once a month orally. These are phase 3 trials, which I believe may have already started. So the Empower 022 trial will be looking at cisgender women and adolescent girls in sub-Saharan Africa, and the Empower 024 study will be looking at MSM and transgender women. And Dan, you'll let me know, right, if there's questions. I'm sorry, I can't see the chat. So next they presented, so they also talked about Islatravir implant. So Islatravir, because of its long half-life, now it's being studied in the form of an implant. And and what they found, this is a study that had been previously published, and what they found is that the PK threshold for Islatravir in implants maintained for months. So they looked at a 62 milligram implant. On the left here, the islatravir triphosphate concentration. Here's the PK threshold in the dotted purple blue line. And you can see that the levels of islatravir maintained for a very long time, even at the point of the removal of the implant. So it will continue to release through 52 weeks. And on the right here, when they did simulations for a year, even at the year mark, there was still sufficient levels of islatravir triphosphate above the PK threshold. In fact, projected time was projected to fall below 0.05 until 60 months. So all of this sounds very interesting, and you can see there it looks like a little toothpick, but it's very interesting. It's very exciting to think about what could come in the future. And these implants are also in development for along with TAF and cabotegravir.
Gabe, of note, I think Ryan had typed in the chat pod that I think they just recently started the trials. So
Empower trials. Okay, yeah. Thanks, Ryan. So this is now another agent that was discussed at CROI. This is Lenacapavir by Gilead. So this is a first-in-class, long-acting HIV capsid inhibitor. So this is a small molecule which disrupts the functions of HIV capsid. And by the way, let me take a deviate for a second just to say that there were some really interesting, excellent talks on capsid imaging and the viral cycle, and I hope you all get to watch Pamela Bjorkman and Hans Kreuzmann. They did a great job of describing some of the recent research in that particular area of the life cycle and things that we're learning even now. So again, this is a small molecule which disrupts the functions of HIV capsid protein. It has high potency with antiviral activity at very low dosages, similar to islatravir in the picomolar range, and no cross-resistance with approved drugs. It has a very low in vivo systemic clearance, and it has slow release kinetics from subcutaneous injection sites, which is great because it can be used again for long-acting PrEP. So again, these are data that have been presented previously, but by way of background, basically you can see here on the left, this is the mean change in HIV RNA after a single subcutaneous dose of lenacapavir at different dosages. And you can see that they're all pretty good and sustained even 10 days out. And on the right, you can see the pharmacokinetic, this is a single dose of lenacapavir at 300 milligrams in the blue and 900 milligrams in the other colors. And and basically it was found that, so yeah, potent viral activity with a single subcutaneous injection with maximum HIV RNA reduction was seen at mean concentrations of greater than 4.4 nanograms. And as you can follow the curve here in the graph on the right, you can see that the lenacapavir actually had sustained its of the target at 6 months. It's when they start to go down. So really allowing for the use of lenacapavir 900 milligrams every 6 months in phase 3 treatment and prevention trials, given its half-life. So there's a lenacapavir prevention trial enrolling in women. This is the women's HIV prevention study, which is randomizing to lenacapavir and against FTAF and against TDF-FTC. And they'll be comparing that to non-PrEP background HIV using recency assays and historical HIV incidence. This is a study that's going to take place in South Africa, Kenya, and Uganda. And in addition to this, there is another trial planned in MSM and transgender women versus daily TDF-FTC in the USA and Africa. So these are fresh data that were presented at CROI from Dr. Beckerman at Gilead. So, and this is the data from the rhesus macaque model, and it looked good. So the graph on the right showing the percent aviremic and the three colors in gray, placebo in this lighter purple. Here they used not lenacapavir, but an analog of lenacapavir called GSCA1, which is commonly used in the animal models. And in the darker purple, the higher concentration of the lenacapavir analog. And there was a significant risk reduction with the lenacapavir analog versus placebo, 86% and 96% for low and high dose groups. They did make a mention of the fact that the infections that did happen in the intervention groups occurred only after marked compound wash, 9+ weeks post-dose. Next, talking about cabotegravir now. So this is a long-acting integrase inhibitor. Many of us may have heard of it by now. Very similar in structure to dolutegravir. It has several favorable attitudes for PrEP, a high genetic barrier to resistance. Again, PK profile similar to the two that I just mentioned, 21 to 50 days. It can be once daily oral, but it can also be used in long-acting injectable forms using nanosuspension formulations. So this is the study HPTN083 was presented last year by Rafi Landovitz from UCLA, and this was a double-blind placebo-controlled trial looking at long-acting cabotegravir against TDF-FTC. And as you can see in the study schema there, that people were randomized to either with an oral lead-in and then randomized to TDF-FTC or to cabotegravir injectables every 2 months for approximately 3 years. And there was also an injection placebo, and there was also a pill placebo. This enrolled 4,570 MSM and transgender women at 43 sites in Argentina, Brazil, Peru, the US, South Africa, Thailand, and Vietnam. And these were results that were previously reported. So this is, and basically showing that it's highly effective for MSM and transgender women. There were 52 infections in over 6,000 person years of follow-up. And as you can see on the left, there were 39 infections in the TDF-FTC arm and 13 infections resulting in an HIV incidence of 1.22 and 13 infections and incidence of 0.41, which is significantly lower. And in fact, in the diagram on the right, the hazard ratio excluded the superiority threshold, so suggesting that it's superior to TDF-FTC. So at CROI, Rafi Landovitz presented a laboratory analysis of the HIV infections that occurred in HPTN083. So here they did a post hoc characterization of the 58 cases of HIV infection in HPTN083, and they characterized these using concentrations of cabotegravir and tenofovir in plasma and tenofovir diphosphate in dried blood spots by liquid chromatography mass spectrometry. And they used the 4th gen assay and the genius discriminatory test and RNA assays and drug resistance testing on samples that had HIV RNA greater than 500 copies per mil. And they also broke down the infections by groups. But I'm good. Okay, so in the post hoc analysis, they actually found one incident infection in the cabotegravir arm that was they found that they had to, and then there was an additional baseline infection. So the number of incident infections actually got lower in the cabotegravir arm, although it didn't make that much of a difference. So it went down to 12. So the incidence in the cabotegravir arm down to 0.37 compared to 1.22, and the hazard ratio was unchanged. So there were 12 incident infections that occurred in the cabotegravir arm. 5 had no recent cabotegravir dose, suggesting that these occurred during the pharmacokinetic tail. So this is the period after which, there is no more cabotegravir in the system. And this is a particularly worrisome, I think, concept and idea for folks is, what would happen when people don't come to the clinic regularly, and because they would be at risk for infection. So and this graph just basically shows, so this is an example. This is one person, one case before. The orange dots are the concentration of cabotegravir, and the green lines are the injections over time. And you can see that after the last injection, participant didn't get administration again, and then he posted positive. 3 of the incident infections occurred in the oral lead-in phase, and 1 had no cabotegravir detected. And as you can see here, actually the 2 cases had INSTI resistance-associated mutations with integrase inhibitors, including the Q148R and the E138E Merck. And 4 occurred despite on-time cabotegravir injections and targeted cabotegravir concentrations. And the evaluation of correlative protection is ongoing in these individuals. Which, again, taking all this into account is still pretty good. I mean, for over 4,000 individuals, it's obvious that there was a great reduction in the incidence of infection, but it is curious to know what happened in these individuals. 5 of the 16 infections in the cabotegravir arm had integrase resistance-associated mutations, and 1 also had an NNRTI resistance-associated mutation. 1 actually had NNRTI mutations only, and 1 had NNRTI along with NRTI. So unclear if transmitted drug resistance, or perhaps some of these individuals had exposure to antiretrovirals in the past. And so this is the conclusion that they made is, so another point that they made is that cabotegravir long-acting can delay the detection of infection using standard HIV testing algorithms, especially the 4th generation assay. Here in our cohort studies and in the PERC study and the early test, we're so used to having the luxury of doing the qualitative RNA assay that forget that for a lot of these PrEP trials, they're not using that. They're using 4th gen assays, and they make it a point. He made it a point to say that they'll be using viral load testing as a primary screen going forward in the open label extension, because of the difficulty sometimes in characterizing a new diagnosis quickly enough in some of these PrEP trials. And he also made it a point to mention that the oral lead-in will be optional in the open label extension.
So Gabe,
Yeah.
just letting you know, there was a comment from Edie Seville about that she's already seen one patient who's had some resistance to cabotegravir, at least based on Stanford database. And so I think kind of citing some concerns. And then Lucas was asked whether or not he's seen any cabotegravir resistance, and just replied that there is cross resistance with other INSTIs. So if someone's had INSTI resistance, they may have cab resistance as well. So I didn't know if you wanted to comment on that.
Yeah, yeah, and it's going to be important. This isn't going to be a panacea. There's going to be resistance out there, and that's where the importance of coming into the clinic and the importance of proper diagnosing and proper testing done quickly will become crucial to identifying potential problems. So, clinic flows will need to take this into account because there's going to be maybe a few, but still not a small number of pre-existing resistance and even acquisition of resistance. Okay, so this was a mirror study to HPTN-083, but this was in women. And so this was PrEP with long-acting injectable, highly effective for women. This was the protocol, study protocol looked very similar to the HPTN-083. Two groups, randomized, there was an oral lead-in, and randomized to the injection every 2 months versus the TDF-FTC. And what they found was there were 38 HIV infections in 3,223 women with a median age of 26 years. This is in Botswana, Eswatini, Kenya, Malawi, Uganda, and Zimbabwe. And they found that there was a dramatic reduction in the HIV incidence, 1.86 in the TDF-FTC arm versus 0.2 in the cabotegravir arm. And that was non-inferior compared to the TDF-FTC. So 36 infection is not a small number. And when they looked at the tenofovir plasma concentrations, looking at those who had more than 40 nanograms in their sample, so suggesting that they had been taking the tenofovir, they saw that the adherence wasn't great. So 60% at week 4, which dropped down to 34% in week 57, which and this echoes what has been seen previously in a few of the other seminal PrEP trials in women, like Voice and others. So, however, the cabotegravir injection coverage remained pretty good, 94% at 6 months and 90% at 30 months. Both products were safe and well tolerated, and there was no discontinuation due to injection site reactions and similar pregnancy outcomes. And this is another self-report from quote from a 17-year-old young woman in the Eastern Cape. And I'll let you go ahead and read the quote. So clearly there's a role for these long-acting agents in a need in the community. All right, so neutralizing antibody CO1 of HIV picture with VRCO1 targeting the C4 binding site. And so we had known before that VRC01 can prevent HIV shift transmission in animal models, but the AMP trials were the first to actually, this was a phase 2b proof of concept trials to test the efficacy of VRC01 to prevent HIV acquisition. So these were not presented at CROI. These had been presented just at R4P, but recently. So 2 harmonized protocols. This is by HVTN and HPTN. HVTN 704, HVTN 085 was in MSM and transgender women, and HVTN 703, HVTN 081 in sub-Saharan African women. This is a placebo-controlled trial of the VRC01 monoclonal antibody, the broadly neutralizing antibody, every 2 months by IV, and both trials opened in April, May of 2016. And VRC01 was basically, it did not show overall prevention efficacy. It was poor. It only showed an overall prevention efficacy of only 18.1%. However, it was able to neutralize sensitive viruses, which was important. This is on the left, the prevention efficacy broken down here. They broke it down by viruses that are like the viruses that are easily neutralizable, less than 1, medium neutralizable, 1 to 3, and fairly neutralization resistant, greater than 3. And what was interesting is that for those who had, it was able to protect against individuals who were infected with highly neutralization susceptible viruses. So that was interesting, and that was good to know. In vitro neutralization assays can predict the outcome, and viruses from the placebo arm. So in the placebo arm, viruses were tested along with other BNAbs, and when you combine other BNAbs, the triple combination can achieve coverage of 90%. So the fact that we would have to combine highly active monoclonal antibodies is not too surprising. But the fact that even a single antibody demonstrated some level of prevention efficacy in this passive immunization model was a very interesting proof of concept. And reminiscent of a finding from an HIV superinfection study that was done long ago. This is in our work with Davey. When we presented our superinfection data, we looked at the neutralizing antibody response in a case control study of people that were superinfected and those who were not superinfected or monoinfected. And what we found in our study, in these 2 graphs, you can see the neutralization susceptibility to 2 highly neutralization sensitive viruses, NL43 and SF162. And individuals who would become superinfected had a lower neutralizing antibody response to tier 1 viruses. And we did this study, almost as to study or to try and do an analogy for vaccination, for the people that are mono-infected are having the same amount of exposure, but for whatever reason, they're not getting reinfected a second time. Is there something different about their neutralizing antibody response? And we even posited in the paper that the neutralizing antibody response of mono-infected participants may have been sufficient to decrease susceptibility, at least to tier 1 viruses, at least to highly susceptible viruses. Of course, that wasn't entirely true in the superinfection arm. There were still some people that mounted some level of activity against SF162 and still got superinfected. But overall, the finding of the AMP studies I found interesting because they actually do seem to confirm something that we had been internally discussing in our group, like, hey, maybe even a moderate amount of neutralization against tier 1 viruses is enough to protect. Maybe it was. So the summary of the AMP studies: proof of concept that long-acting BNAbs can prevent HIV acquisition. The in vitro susceptibility to VRC01 influences preventive efficacy. So unfortunately, to VRC01 alone, it's only active against against highly neutralization sensitive viruses, which is only 30% of circulating strains, but that's not zero. A neutralization titer or antibody concentration in serum was established as a biomarker of protection, which is great. And viruses from VRC01 cases were more resistant to VRC01 than viruses from the placebo cases, which suggests already immunologic pressure even from that single antibody. So as I said previously, it's becoming clear that multiple BNAbs will be needed for optimal prevention. And I'd like to end, well, this is one of my final slides, is I didn't get to discuss anything. I didn't discuss anything on COVID. There was a lot of talks on COVID, but there was a lot on HIV prevention, so I focused on that. But one thing that was brought up is how the HIV BNAb research field facilitated the rapid development of the SARS-CoV-2 NAbs. So as you can see here in the timeline, was around 10 years ago, the development of technologies for single B cell sorting and single B cell cloning and sorting is what allowed the generation of this generation of BNAbs. And so it's been 10 years. But in that time frame, I mean, now looking at just 1 year, it took 1 year to develop the neutralizing antibodies against COVID-2. So the isolation of SARS-CoV-2 antibodies for safety and tolerability between March and May of 2020, and already the clinical trials that are ongoing now. So pretty impressive. And I'd just like to end, I would invite you to the Martin Delaney presentation at CROI. Was fantastic. So Greg Gonsalves from Yale and Fatima Hassan from South Africa did they both longtime AIDS activists, and really highlighted the importance of vaccine nationalism and how it could hurt the efforts at the eradication of the pandemic. And so vaccine equity, #no vaccine apartheid. I would encourage you to check out their presentation. Was really lively and very very timely. And I think that's about all I have.
Okay, great. Thanks, Gabe, for the wonderful talk. There were a few comments that we didn't talk a little bit about, and I'll start from the beginning. I think Jill had mentioned a comment about FTAF. I think this was FTAF versus the regular TDF in regards to PrEP. And she said, unless the females were having anal sex acts, they really should not have been switched to FTAF since not approved yet, unless if the female data was capturing transgender women. And so this had to do with the, the FTAF versus FTDF. Do you remember if the female data was capturing transgender women or not?
I don't know. Jill, are you referring to the Prevenir study? I think it was
no. The just the Hoover trend data, which showed it was about the FTAF switches.
Okay.
And it was surprising that there weren't that many. I think it was like a little under 2,000 switches to FTAF for females. And this is just AQAVI data. So yeah. I think it's whatever is on their administrative, whatever legally is written administratively. So it could capture some trans women. But I did think it was interesting that was there, to begin with. And I was just making the comment about that.
Yeah. I don't know.
I think the more interesting part of that was actually that only it was around 7% of females, again, whatever that means, are being prescribed PrEP right now. That was what the trend data showed, which is, just so low. So I had to cover this for something else, and I was very struck by this data, and I just wanted to make the comment because the risk, they said that what was associated with the switch to FTDF included being male, but that makes sense since technically females, unless they were only having anal sex, they really shouldn't be getting
it. Yeah. In fact, yeah, probably didn't even need to be reported as
Right.
given.
Sorry if that was sorry, I was not very helpful with that comment. I apologize.
No, thank you for commenting.
There was also a comment from Gigi in regards to Islatravir that they need to study it with contraception too, and with the comment that it will need to last 3 years. And I guess Gigi or Jill had also put out to Gigi that there was, I think, something presented at CROI on long-acting co-formulated biodegradable implant for HIV prevention and contraception. But I didn't know if you had any thoughts about Islatravir and contraception.
Yeah, it is being looked at yes, that's being looked at. The field is really cognizant of the bundling of agents, in particular contraception and HIV prevention for women. So it's really that's key, and clinical trials are really looking at testing that for sure. I'm not too familiar with the study that I didn't see that one.
Yeah, it was
presentation
just it was a science spotlight. I'd urge anyone interested in this field to look at it. It was very cool data. They looked at Islatravir and TAF looks like it will be a little bit less stable. So Islatravir may be the best way to go, but it was just looking at yearly data, or yearly length of time, Gigi. And these were in vitro studies, so we're pretty early on in things, but yeah, so it was a science spotlight, and there's a video with it, so I urge people to check it out if they can.
Great. Any other questions or comments? I think the last question I'll pose to you is, just given that we have all of these new things that may be coming out, are you concerned that once something, will any of these agents then make other agents obsolete, or what do you foresee as the future of even things like TDF or FT or TAF in the future, providing we have some of these longer acting agents?
Yeah, so several people at CROI had opinions regarding this, and I tend to agree with this notion of kind of the more the better. The more choices are out there, the better. When it comes to HIV prevention, there isn't going to be a single shoe that fits everyone. And some people will like daily oral PrEP, and some people will prefer to have long-acting injectable that lasts 3 months or 6 months. And some people will capitalize on bundling of HIV prevention and contraception. So I think the more choices out there, the better to fit the diversity of people's lifestyles. So no, I think there will continue to be a role for older agents as well.
Okay, great. If there aren't any other thoughts or comments, I'll go ahead and end our conference today, and wanted to thank Gabe for excellent presentation and overview of kind of what's to come for prevention, and hopefully we will see you all next week. Thanks, and have a good weekend.
Thank you.
Thanks, Gabe. That was great.
Thank you.